Oh, well, Stella wanted to say hi for this video. Say hi, Stella. Mm. Okay, over there. All right, so this is Anatomy, Chapter 10, Part 2. Continuing to talk about the proteins associated in the sarcomere. The main proteins are the thick and thin filaments. The thin filaments are made of actin, and actin are like beads or bead of pearls strung together. You can see them here. The thick filaments, like I said, they have those myosin heads, which I think look like golf club heads. All right. Okay, so also on the actin, we have these little, I think it's blue or black dots that are referred to as the myosin binding sites. And you guessed it, the myosin heads will bind there, but not right away. You have these other regulatory proteins called troponin right here and tropomyosin that essentially block those myosin binding sites until a specific time, okay? The reason why they're blocked is because if they were open all the time, your muscles would be contracting all the time. And we don't want that, only at certain times that is highly regulated. So what happens is that troponin-trimyosin complex, when activated, will slide down into the gutters or move out of the way to unblock those myosin binding sites. And what triggers them to move out of the way is an ion called calcium. Calcium will be released into the muscle cell and go to those areas, attach, and cause those proteins, the troponin and myosin, to move. Once they move, those binding sites are exposed, and you can see here the myosin will then, those heads will bind right to those regions, okay? So the myosin is grabbing the actin. All right, you can see another image here. This is the actin, this is the myosin. Myosin head grabs it, okay? And what happens, remember, we have them every other. So let's say this one's myosin, my fist here, and this is actin. It'll grab on, and it will pull, pull, pull. If both my fists are myosin and I have invisible actin above me, it's pulling, it's pulling, it's pulling. The fibers slide over one another. The myosin actually brings the actin in closer. So the fibers slide over each other, essentially shortening that sarcomere. Okay. So if this is the sarcomere to start from my wrist to my wrist, and myosin pulls it in, you can see that that width is shortened. It's called the sliding filament mechanism. Sliding. The filaments don't get shorter themselves, but they slide over one another. Here's what it looks like under a microscope, and here is the schematic view of what's happening. Here is our two sarcomeres together, okay? In this image, Z disc to Z disc, okay? And all the zones, the H zone, A band, I bands, okay? And when a muscle is relaxed, this is what it looks like. But when the muscle starts to contract, essentially when that troponin and myosin move out of the way, the myosin grabs onto the actin and starts pulling it, sliding it over each other. You can see that sarcomere is shortened. Okay? This is a maximally contracted muscle. It's a 4-step process here that allows that to happen. The first step is the hydrolysis of ATP. ATP we associate with energy, okay? That energy, essentially, when it's hydrolyzed, remember hydrolysis, water breaks. Water comes in, breaks that ATP, and separates it into ADP plus a phosphate. Essentially, you could write this down to it cocks that gun, getting ready to shoot, okay? That's what happens. Step 1. So now that myosin head that is a cocked gun ready to fire, okay? It's full of energy, ready to go. Step 2, attachment. And we also note that calcium is the little, I think, a little purple things in this diagram. So calcium has opened up those sites. Now the second step is attachment. Those myosin heads bind to the actin. Third step, myosin cross bridges rotate towards center of sarcomere. It's called the power stroke. So myosin heads have attached, okay, and now they're pulling, pulling, pulling to shorten that sarcomere. Oops, undo. Detachment is the fourth step in which the myosin heads let go. It's like in tug of war when you let go of the rope, okay? And the reason why they let go is a new ATP molecule binds to that myosin, okay? That triggers it to let go, okay? And what will happen, it will drop that rope, drop that actin, and find another region to attach to keep pulling. Keep pulling, drop, pull, drop, pull. That will happen as long as there's calcium and ATP in that area. Essentially, that muscle will contract as long as you have available energy, ATP, and as long as you're receiving that message to contract, thus the calcium. And this is all of it together. It is a cycle, like I said. It'll keep going as long as there's calcium and ATP. And I would like you to watch this video. It's linked on the online notes, and it'll be a very good overview of that process. Okay? When your muscle contracts, okay, the sarcomere shortens. We talked about that. We'll take a look at this graph. This is the sarcomere length, okay? And this is the tension of the muscle. When the sarcomere shortens, it produces a tension within the muscle. You can overstretch a muscle or understretch a muscle. Okay, that's what this is showing. So overstretched muscle would be those myosin actin are way too far apart from each other. So essentially, the myosin's having a hard time even grasping the actin. In an understretched muscle, the fibers are too far over each other, so it can't produce enough efficient tension in the muscle to get your best results. Essentially, is what that means. You want to keep it in the optimal length to produce the best results for muscle contraction. And that is the end of part 2.